We present a method for live image analysis of LDL cholesterol uptake into various human cell types. This allows to screen for compounds that modify the transport of LDL cholesterol.
The main advantage of using live cell imaging for LDL influx quantification is that it allows monitoring cell morphology throughout the assay duration, so potentially cytotoxic compounds can be detected.
To begin, aspirate the media off the previously prepared cells and wash the cells with 5 ml of Dulbecco's phosphate buffered saline. Aspirate the DPBS. Next, to detach the cells from the 100 mm dish, add 1.5 ml of 0.25% trypsin EDTA for the HepG2 cells, and add 1.5 ml of 0.05% trypsin EDTA solution for the HK2 cells or the HCAECs. Incubate the plates at 37 degrees Celsius for 4 minutes. Then, neutralize the trypsin by adding 3 ml of complete media for the HepG2 and HCAEC cells. Use 3 ml of the trypsin neutralizing solution, comprised of DPBS with 5% fetal bovine serum or FBS for the HK2 cells. Transfer the cells into 15 ml conical tubes. Then, centrifuge the tubes at 250 G for 5 minutes. After the spin, aspirate the media and resuspend the cell pellet in complete media. Filter the cell suspension gently through a 40-micron mesh strainer to break up cell clumps. Count the cells and plate them at an optimized density in a